Hello, my name is Maren Elise Rognerud. I'm a master's student at NTNU, the Norwegian University of Science and Technology, and I'm writing my master thesis at the Water Power Laboratory. In this presentation, I'm going to present the article that I have been writing for the conference at TTL Kathmandu University. Unfortunately, this could not be done in Kathmandu, so thank you very much for watching this presentation anyways. My topic is how you can reduce gas supersaturation from hydropower plants, which is a very not too well-known problem, but it has very large impact on the biodiversity in rivers where it actually occurs. So this is me, and thank you for listening, and let's get started. So the agenda for this presentation will be first to explain what total dissolved gas supersaturation is. Then I will move over to explain how it can occur in hydropower plants and what the consequences of that would be. After that, I will try to explain how ultrasound can be used to enhance the degassing process and how to remove air from the water. After that, I will present some experimental results to illustrate how this could work. These are small-scale experiments, and this can be used to suggest further experiments later. Finally, I'm going to conclude my work and maybe see what this could be used for later. Total dissolved gas supersaturation is something that could happen if the air content in water is higher than the solubility threshold in water. So the general rule for air solubility in water is that the solubility increases with increasing pressure and decreasing temperature. So at high pressures, water is able to hold more air than at low pressures. That would be because the molecules are compressed together, and that allows for more air to stay within or between the water molecules. Also, at decreasing temperatures, the molecular vibration is lower, so this also allows for more air to be stored in between the water molecules. This can be seen from this graph as well, where low pressure gives a low solubility threshold, and also lower temperatures will increase this solubility threshold. So for high pressures and low temperature, you can dissolve more air in the water. So then total dissolved gas supersaturation is what happens if your water contains more air than it would is able to hold on to. So you have more air dissolved in the water than the solubility threshold. This might happen, for instance, in a hydropower plant. As we know, the pressure in the system is increasing the closer you get to a hydropower plant. So in the pressure shaft, the pressure is really high. If you get air in through brook intakes or other places in the system, this air will be transported into the pressure shaft, and the high pressure will allow for this air to be compressed in through the water molecules and course, the pressure is higher, so the solubility threshold is also higher. At this point in the pressure shaft, the water is able to store more air than it is at the ambient pressure. So what happens is when this water is moving through the turbine and out to the lower reservoir, it will suddenly be exposed to atmospheric pressure, which is a lot lower than the pressure in the pressure shaft. And when this suddenly happens, the solubility threshold is much lower than what it was in the pressure shaft, and air bubbles will start to form because the water is getting rid of all this air. And that is how you get supersaturated water downstream a hydropower plant. When this phenomenon occurs, you will have air with or water with a lot of air bubbles floating in the river, and as the water is moving downstream, more and more bubbles will form and make the river look milky white, like you can see in this picture. The consequences of total dissolved gas supersaturation in hydropower plants can be severe for the fish and other aquatic species that lives in the river. When the dissolved gas starts to come out as bubbles in the water, this is happening over long stretches of the river. If this happens, the bubble formation, as the water is moving through the gills of the fish, these bubbles might be transported into the body of the fish, to the eyes, as you can see in picture B, or other parts of the fish. So in this first picture, you see number A, which is just a normal fish, and number B, which is a fish who is exposed to total dissolved gas supersaturation. And this is a very serious condition for a fish, and it can kill them if it's going or if it's staying for a longer period of time. So now we're moving on to looking at one solution for how to remove air from water to avoid total dissolved gas supersaturation. And this solution is by applying ultrasound to the water volume. Ultrasound is sound waves with frequencies above 20 kilohertz. Ultrasound can be divided into high power, low frequency ultrasound, which transfers high levels of power into the water and also needs a higher power to produce the ultrasonic waves, and low power, high frequency ultrasound, which needs less power to be produced and also has lower frequencies. So ultrasound can also be described by sinusoidal pressure waves with an amplitude PA and given frequency. These ultrasonic waves can be used to reduce the gas content in water. And the way this works is, as you can see in this figure, the pressure waves are represented with compression phases marked with a C and rarefaction phases marked with an R. So at the compression you have high pressure in the water, and at the rarefaction you have low pressure in the water. And the process that is making the air removed from the water is a process called rectified diffusion, and this happens when you get cavitation bubbles in the water at the rarefaction phase of the ultrasonic wave. So at a rarefaction, a cavitation bubble will occur in the water. A small bubble, as you can see here, that will occur when a rarefaction phase is appearing. This is happening because the rarefaction pressure is lower than the cavitation threshold pressure in the water. So when this cavitation bubble is compressed during the compression phase, some gas molecules will diffuse out through the bubble surface into the water over the area of the bubble surface. Further, when you get into a rarefaction phase of the ultrasonic wave, the pressure outside the bubble will be lower, so the bubble will expand because it has more space. And when this happens, you have a larger area of the bubble. You have a low pressure inside the bubble, and this allows for gas molecules to diffuse from the water into the bubble to fill up the bubble with air. Because gas is compressible, when a new compression phase is coming up, the bubble is decreasing in size again, and some gas will escape through the surface, but not all, and the surface area is still small. So when then a new rarefaction phase comes, more air is transported into the bubble again, and the bubble is a little bit larger than it was before. So you have two effects working here. First, it's the area effect that allows for more air to enter the bubble when you're in a rarefaction phase compared to what is leaving the bubble during a compression phase. And you also have an effect that comes from the fact that the air has less resistance when going from a high density fluid like water into a low density fluid like gas inside the bubble, and it's more easy, or the gas can move from the outside of the bubble to the inside of the bubble more easily than the other way around. So all this together makes this bubble grow piece by piece, working like a pump almost. More air is transported into the bubble compared to what is leaving it. So when you finally get enough air inside the bubble, the buoyancy force will be strong enough to lift the bubble up and out of the water. And this is how ultrasound is used to enhance degassing of air from water. So now that we understand how the degassing mechanism is working with ultrasound, it's time to present the setup that was used for the experiments. So in this picture, you can see two different glass bottles. The first one, just this one, is having a possibility to cool the water. It's water inside. So this water will be cooled, and underneath it we have an ultrasonic transducer. So in this container you will have water and ultrasound, which is applied to the water. From the first container to the second container, you will have tubes to circulate the water. So the water will be circulated from the first container through a pump and into the second container. And the purpose of the second container is to make the measurements. And the reason why these two processes, the sonication with ultrasound and the measurements are separated, is because the ultrasound might be causing problems to the measurements. So that's why they are separated. When the water enters the second container, we'll take measurements there, and the water will flow back into the first container again. Also, in the first container, oxygen is supplied to the water through a glass tube, which you can see here. So oxygen is just bubbled into the water at atmospheric pressure, and this is actually good enough to saturate the water or supersaturate the water. So the process of doing the experiments would be to first start the pump, circulate the solution between the two containers, the first and the second one, and bubbling oxygen into the water. When the water is reaching saturation of 18 milligrams per liter, which is about 230% supersaturation, you will be ready to start the ultrasound. So we're saturating the water to 18 milligrams per liter of oxygen. We're using oxygen only because it's easy to measure. Then the oxygen bubbling is stopped and the ultrasonic transducer is turned on. And then as the ultrasound is running, the measurements are taken once every minute until the saturation level is back to the initial saturation level, which is somewhere between 7 and 8 milligrams per liter. This experiment is repeated for many different ultrasonic frequencies, all from 24 kilohertz and up to 1140 kilohertz. And it's also done without the ultrasound to compare the degassing process to how the water is degassed without using ultrasound. The results of these experiments are shown in this graph. We can see the different frequencies that have been tested from 24 kilohertz and up to 1140 kilohertz, and NU stands for no ultrasound. So these are experiments without ultrasound applied. And we can see that the most effective frequency was 24 kilohertz, which was able to go down to 8 milligrams per liter in about 20 minutes. The other frequencies spent longer to get down to 8 milligrams per liter. One other thing that we can observe from these results is that you have very good degassing efficiency for 24 kilohertz. Then it's less good for 40 and 580 kilohertz. And then it's getting better again for 860 and 1140 kilohertz. These are some interesting phenomena that occurs because of different effects that are introduced by the ultrasound. One thing is that you have larger bubbles at lower frequencies, but at the same time you will have a larger number of bubbles for higher frequencies. And because of this, you can end up getting a better degassing efficiency for low frequencies and high frequencies, and a less good area in between these two, which is a very interesting phenomena that occurs. So yes, these are the results. Finally, moving through the conclusions, we have seen that the fastest completed degassing of 1.2 liters of water from 230% supersaturation with frequency of the ultrasound of 24 kilohertz and a transmitted power 134 watts is measured to take about 20 minutes. This is quite a long period of time, and in a hydropower plant, of course, you would need to have shorter exposure times than this. This is also a very small scale experiment, so in order to apply this phenomena in hydropower plants, we would need to scale up a lot to see if it's actually possible to make or to use this concept to degas the water. But the most important thing is that we have seen that it actually works to degas water by using ultrasound, and it's quicker than not using ultrasound. The next point is the exposure time of the ultrasound in these experiments. It's been in the order of 20 minutes. In a hydropower plant, you would get down to in the order of 1 second of possible exposure time. So this is going to be a challenge. You might need to increase the power to increase the degassing efficiency. But this also needs to be tested in maybe a small scale model. Finally, the next step would be proving how this concept is working in flowing water. So we have proved that this is working in a small container when the flow rate is really, really, really low. But the next thing would be to get a scale up to a larger amount of water and see what happens when this water is flowing through the ultrasonic field. So that was everything from me. Thank you very much for watching.